All right, guys, assalamu alaikum. My name is Rasoul Turko, and today we're going to talk about amino acid synthesis. So the amino acid synthesis is part of 5 different lectures that you guys will be taking in MOL 2, where you'll be talking about the synthesis, degradation, and one second, this is synthesis, yeah. Synthesis and metabolism of amino acids. So in order to get the full picture, you guys will have to go over those different lectures, and what's good about it is that a lot of the concepts will be repeated throughout the 5 lectures. So if you guys get all you guys have to do is get the basics, and then multiple stuff will be repeated over and over the span of the 5 lectures. Okay. So our objectives is going to be, we're going to talk about glucogenic and ketogenic amino acids. So so you guys already talked with Dr. Raja about when we break an amino acid, we'll get ammonia, and then we get a carbon skeleton, right? So this lecture is basically, we're going to be talking about the carbon skeleton part of the amino acid, and what makes it a glucogenic or a ketogenic amino acid. And then we're going to talk about that carbon skeleton and how it gets broken down and what it gives us as product. Okay, so amino acids. So this is an introductory slide where we talk about, again, so the amino acid was broken down into, and we take the amino out, the amino group out, and then we will have a carbon skeleton. So here we'll have a carbon skeleton, or AKA alpha keto acid. So it's called an alpha keto acid or a carbon skeleton. And that carbon skeleton is broken down to give us energy. So how does it give us energy? How does it get broken down is what this lecture is about. Okay. So again, yeah, so it's going to be used as a fuel source, or to give us other molecules that will give us energy. So now we're talking about glucogenic and ketogenic amino acids. So the amino acid, when it gets broken down, it can give us either it can be a glucogenic or ketogenic. How? A glucogenic means that amino acid, by it getting broken down, it can give us products that are in the TCA cycle. If you guys remember the TCA cycle, where we have different substrates that would give us ATP. Yeah, so amino acid gets broken down into either pyruvate or some of those substrates in the TCA cycle. So that's what makes an amino acid glucogenic. A ketogenic amino acid is an amino acid that gets broken down into acetyl-CoA or its precursors, or which is acetate or acetyl-CoA. Okay. Or it can be both. It can be an amino acid that can give us both a TCA cycle substrate and acetate or acetyl-CoA. Okay. So glucogenic, again, so it's an amino acid that gets broken down to give us intermediates of the citric acid cycle, which are 5. They are pyruvate. Sorry, 1 second. Yeah, so it's pyruvate, alpha ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. So those lectures, this one and the one after, is going to be a lot of recall, a lot of stuff that you guys have to memorize. What me and Amr tried to do is we tried to make that memorizing as simple as possible. We tried to put mnemonics and stuff that would make it easier for you guys to memorize. But this is one of those slides where you guys have to memorize those different products that would make a amino acid glucogenic. Okay, so it's pyruvate, alpha ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. Those are substrates that would give us glucose in the end. Okay, those all give us glucose, which make it a glucogenic amino acid. Okay. Ketogenic amino acid is, again, an amino acid that gets broken down into acetoacetate or its precursor. So acetyl-CoA or acetoacetyl-CoA. Okay. So how do we know if it's how do we know if this amino acid is glucogenic or ketogenic? I have this little mnemonic that made me remember it. So kill, as in K, is ketogenic. ketogenic amino acid, and then two L's. I is lowercase because it's not part of the mnemonic. So I remember it as KLL. L here as in leucine, and L here as in lysine. Okay. So the way I remember them is I remember what is ketogenic, which is those two, leucine and lysine. I remember what can be both, which is another, it's another mnemonic that I have here. And if it's not ketogenic and it's not both, then it's going to be a glucogenic. Okay. Because glucogenic is there's a lot more amino acids that are glucogenic than ketogenic or both. Okay, so yeah, leucine and lysine are the only exclusively ketogenic amino acids, and they are found in proteins. And those fatty acids can be converted to those amino acids can be converted to fatty acids and ketone bodies, which means lipids. So those still give us energy, but they give us energy in the form of lipids. Okay. Okay, so this slide you probably guys went over before. So how do we classify amino acids? We classify them as essential and non-essential. Essential amino acids are in the form of PVT to MOL, which means those amino acids we can't make, so we have to eat. So PVT to MOL is the essential amino acid. And then it can be divided into glucogenic, ketogenic, or both. Okay, so again, we said ketogenic is this mnemonic, which gives us leucine and lysine, Both is another mnemonic that I have. It's IPTT. Okay, so it's I as in isoleucine, P as in phenylalanine. And what's nice about it is that all the T's, all the amino acids, they're all both. So tyrosine, threonine, tryptophan. Okay, so if you guys get a question and it says which of the following amino acids is glucogenic, for example, you guys will go over, like, is it leucine and lysine ketogenic? No. Is it both? No. Then it's glucogenic for sure. Okay. Okay, so now for location of amino acid degradation. So all this catabolism that happens, which gives us either a ketogenic or a glucogenic amino acid, happens predominantly in the liver. Okay, so most of it happens in the liver. Sometimes it happens in different organs such as the kidney, small intestine, muscles, and adipose tissue, but it mostly happens in the liver. Okay. Here also, Dr. Hannah briefly talked about BCAAs, which you will be taking in later lectures, but she briefly went over it just to tell you that there are branched chain amino acids which get degraded in the muscle. So this for example, like these are basically supplements that people that go to the gym take to give them strength and give them better energy to grow muscle. So it makes sense why they would be broken down in the muscle. Okay, so they are 3 amino acids, and the mnemonic we have for them is I love valine, which is 3 amino acids, is isoleucine, leucine, and valine. Okay, so all you guys have to know for now is that there is such thing called BCAAs, which is branched chain amino acids, and they are isoleucine, leucine, and valine, and they are broken down in the muscle. This is all the doctor talked about so far. You guys will get more in depth about it in later lectures. Okay. Here we have our first checkpoint. You guys have any questions so far? Okay, I'll keep going. If there's a question, just stop me. Okay. All right. So now we went over what happens when an amino acid gets broken down. Okay, so now we're going to look at their results, or what do they give us. So again, this is a slide where there has to be some memorization going on. So there's an amino acids here, and you get their products over here. Okay, we get their products over here. So first of all, alanine alanine gives us pyruvate, and aspartate or aspartic acid gives us oxaloacetate. Those two amino acids or those two reactions happen through aminotransferase, ALT and AST. And you guys took them in MOL 1, I believe. So here there's an alanine, we take an amino group out of it gives us pyruvate. And when we have an aspartate, we take the amino group out of it gives us oxaloacetate. Okay, so pretty straightforward. So these questions would come in the form of like, for example, this is the amino acid, and what kind of product does it give, or like what kind of product does accumulate when this gets broken down. Okay, so again, alanine gives you pyruvate, aspartate gives oxaloacetate, glutamate, sorry, gives you alpha ketoglutarate, asparagine gives you aspartate, and glutamine gives you glutamate. So those last two, so first three happen because of aminotransferase, but those last two happen because of deamination reaction. Okay, so in an aminotransferase reaction, which happened over here up in alanine aspartate, it's called the aminotransferase because we transferred an amino group from one place to the other, from alanine somewhere else, which you guys will be talking later. But in deamination is that we had an asparagine or a glutamine, and we just took the amino acid out. Okay, so and their enzymes are pretty straightforward. So asparagine gets broken down by asparaginase, and glutamine gets broken down by glutaminase. Okay, so they're pretty straightforward. Okay, so this is more in depth about the reaction. So one thing I did is that when I was studying MOL 2, I hated how complex their pathways looked. Even though like we as doctors don't need to go in depth and to know exactly what happens like in terms of enzymes and like chemical structures and stuff. So what I tried to do is I tried to like simplify the pathway and tell you exactly what you need to know. Because at the end of the day, all we care about is the substrate, the product, enzyme, and the cofactor. That's all we care about. So for example, the first one we talked about, the alanine to pyruvate, the enzyme is alanine aminotransferase, okay? And their cofactor is B6. So here I put B6 and I put two different names for it because I know sometimes they try to trick us in the questions by like putting a name that people are not used to. So for example, instead of putting PLP, they put a B6. Yeah, so I tried to put those pyridoxal phosphate, PLP, and B6 are the exact same thing. I wanted to get you guys to know like all the different names so like they don't confuse you in the exam. So an aminotransferase requires a B6, which is why it's right here. So from now on, if you see aminotransferase, it almost always requires an B6 or a PLP. Okay, so this reaction is carried out by alanine aminotransferase, and this is the cofactor. For asparagine, so we get asparagine. We said this happens by a deamination reaction. So asparagine by asparaginase gives us aspartate, and that aspartate, because of that aspartate, goes through aminotransferase to give us oxaloacetate. And those aminotransferase need again PLP or B6. Okay, so pretty straightforward. Here there's a clinical significance for the asparagine. So asparagine is one of the amino acids that the cancers. No, so when someone gets cancer, that cancer needs asparagine to grow. So when a doctor prescribes the patient asparaginase, it metabolizes the asparagine, and therefore we get a lot of like aspartate instead of the asparagine. So when we have less asparagine, that means the tumor won't be able to grow as much. Okay. here for phenylalanine and tyrosine, you guys will be hearing a lot about this reaction in the future. But for now, all you need to know is that phenylalanine, again, phenylalanine and tyrosine are part of the IPTT, which means both, which means they give you a glucogenic and a ketogenic product. So here you see we get fumarate, which is part of the 5 we talked about, the 5 substrates. And then here we get acetoacetate, which is also ketogenic. Okay, so this means that both tyrosine here and phenylalanine are amino acids that give us both. Okay, so what's important here, and you guys will talk about it later, but what's important is that reaction from phenylalanine to tyrosine requires a cofactor, which is tetrahydrobiopterin. Okay, I also wrote that. I wrote that in 3 different forms: so tetrahydrobiopterin, THB, or BH4. I wrote it in 3 different terms, so you guys will know in the exam. So here it would need the tetrahydrobiopterin to become a dihydrobiopterin, which would then yield tyrosine. Okay, so for example, in the exam, if they ask you what cofactor does that reaction require, it requires THB or BH4. Okay. All right. So now for the threonine serine glycine metabolism. So both serine and threonine give us glycine in different ways, and then that glycine gets broken down to give us ammonia and CO2. Okay, and 80% of the times. There's the 20% of the time glycine can be broken down into something that's called a glyoxylate. Okay, and that glyoxylate gives an oxalate, which is basically 80% of what kidney stones have, and that is calcium oxalate. So when we have an increase in glycine, that means that increase in glycine is being broken down into oxalate. So a person with increased glycine would probably have kidney stones in the future. Okay, so this is where it's clinically important again, because glycine, part of its degradation is to give us oxalate in 20% of the time. So this is the significance here. And what's also important is that there's 2 amino acids that can give us glycine in different pathways, and that's threonine and serine. Okay. Okay, so now glycine cleavage reaction, so we're talking about right here, that cleavage reaction happens with the help of 2 cofactors. Happens because of B6, which is again, B6 is PLP, same thing, and happens because of tetrahydrofolate. Okay, tetrahydrofolate which is B9. It requires those 2 cofactors to give us CO2 and H4, to give us ammonia, CO2, and NADH. Okay, and that is by the help of cleavage, glycine cleavage enzyme. So again, I kind of just wrote exactly what you guys need to know from the reactions. So like the cofactor and the enzyme, and that's all you need to know for this slide and for this lecture. So like most importantly is to know that there's some cofactors that we need. What would happen if we lose those cofactors and the enzyme that gives us the amino acid? Okay. Okay. So now for methionine cysteine metabolism. Am I going too fast? If you guys have any questions, stop me whenever. Okay.
Yeah, it's fine. Don't worry. All clear.
Okay. Sounds good. All right. So methionine cysteine metabolism. So here again, so the doctor is telling you that methionine is degraded to succinyl-CoA. And again, we said succinyl-CoA is part of those intermediates here. It's part of those intermediates here, which makes it a glucogenic, okay? So methionine gets broken down into what we call succinyl-CoA. The pathway of it is what we're going to go over here, okay? So what you need to know is that it's required methionine with the help of an ATP, with the help of the ATP, transfers into something that is called SAM, which is S. The S here stands for sulfur. Okay, sulfur adenosylmethionine, which is SAM, and that is a major methyl group donor. So that group, so we need methionine plus ATP to get SAM. That SAM will donate that that methyl or that ATP somewhere else. We're going to talk about it in the next slide. And then after that, we get homocysteine. Homocysteine, sorry. And homocysteine is one of the bad products because it is a metabolite that is associated with vascular disease, with heart disease, atherosclerosis, and the thrombosis. So when there's high levels of homocysteine, that can increase the chance of us developing vascular disease. Okay. So the actual pathway of it, again, I wrote it here and I like highlighted what exactly is needed to know. So methionine, with the help of one ATP, can give us SAM. Okay. That SAM now has that ATP. It has that methyl group. It donates that methyl group or the ATP and becomes SA. Okay. Now this ATP that gets donated here gets donated to a variety of different molecules. For example, norepinephrine. It can get donated to a norepinephrine, for example. And that happens because of the enzyme methyltransferase. Okay, which makes sense because again, the SAM had the methyl group. That methyl group like moved. So methyl transferase makes sense for that reaction. Now that SA then can become homocysteine, and it's important here to know that it becomes homocysteine with the help of adenosine. While producing adenosine, so it produces homocysteine, and adenosine. Now after homocysteine, there's two different pathways it can take. So the first pathway it takes is that if we don't have enough methionine, if there's deficiency in methionine, it goes down this way and produces more methionine. Okay. Now, if the methionine storage was adequate enough, then it would go and produce another amino acid called cysteine. Okay. And what's important here to know is that whichever pathway it takes, it needs different cofactors. So if let's say we didn't have a lot of methionine and we wanted to produce more methionine, we would need two cofactors, which are THF, which again is B9. Like that's a different name for it, is B9, and B12. I wrote here N5 methyl THF because we're gonna talk about it in 2 slides, I believe, that there's different types of THF. There's N5 THF, there's N510 THF, and there's N10 THF. And every type works for a different enzyme. So for this enzyme, homocysteine to methionine, it's gonna be N5 methyl THF, which is B9 also, and B12. Okay. Now, if we have enough methionine and we want to go to cysteine, we would need PLP or B6. Okay. So again, methionine, first of all, with the help of ATP, gives you SAM. That SAM donates the ATP. Usually donates the ATP to norepinephrine, becomes SA. SA produces homocysteine and adenosine, and then that homocysteine either goes to produce methionine with the help of B12 and B9, or goes to cysteine with the help of B6. Okay. So now the cysteine here, what we need to know is that it's made of serine and the sulfur. So it's made from a carbon skeleton that comes from serine, and the sulfur that comes from methionine. And it kind of makes sense because we said here methionine has a sulfur, which is basically in the S here. So methionine gives the S to cysteine. So gives the S, the sulfur, to cysteine, and then it gets the carbon skeleton from serine. And we need to know that the breakdown of cysteine gives us sulfite, which is oxidized and excreted in the urine. Okay, and that's what you need to know from cysteine. Okay, we have another checkpoint here and another question. Which of the following statements concerning amino acids is correct? If you guys can look at it and tell me what you guys think. Anyone has an idea? Is there anyone typing, Zahra?
No, still. Give them a minute. I think they're thinking.
Okay. So what's best to what I do in this case is I try to like eliminate what's wrong. Because when you're eliminating, then you're like, first of all, reviewing different concepts that you have in your head, and like you really like you won't be confused about your answer. You will know for sure that this is the right one. Okay, so let's go over the first one. So they're saying alanine is ketogenic. Is alanine ketogenic? So
Okay,
we
someone
said
typed in the chat. Is it D?
what did I say?
They said like D question mark.
D?
And another one said B.
Okay, so D is the correct answer, yes. Let's see why. So first of all, is alanine ketogenic? So we said ketogenic is there's only two amino acids that are ketogenic, leucine and lysine. So that removes A. B, so amino acids that are catabolized directly to acetyl-CoA without forming pyruvate are glucogenic. That is also wrong. Why? Because amino acids that are catabolized to acetyl-CoA, that makes it a ketogenic ketogenic amino acid. Because acetyl-CoA is not part of the 5 intermediates that we talked about. Okay. Now, if they said the amino acid that are catabolized directly, let's say they said directly to pyruvate, then it would be correct. Okay. So branched chain amino acids are broken down primarily in the liver. So if we said normal amino acids, then it would be correct. But branched chain, so BCAAs, we said BCAAs are broken down in the muscle. Okay. Now, cysteine is essential for individuals consuming a diet severely limited in methionine. Now, why is that correct? Because we said that here, yeah, we said that. So they're saying that this person has low methionine. So if there's low methionine, that means they're going to go through this pathway, come down to homocysteine, and then because there's no methionine here, then the pathway is going to go this way, and we're going to produce more methionine. That will lower the amount of cysteine we have in our body, because cysteine again would only be produced if we have enough methionine. So here, what they're saying, for someone that doesn't have a lot of methionine in their body, we need to eat some parts of food that have cysteine. Okay, and then again, alanine is an essential amino acid for PVT to MOL. Alanine is not part of it, so it's not an essential amino acid. Okay. All right. Any questions so far?
No, all clear.
All right. Sounds good. Okay, so for metabolism of histidine. So the slide in Dr. Hannah's lecture, to be honest, like looked scary. So I tried to like divide it into two parts, because honestly, like there's not like a lot to grasp from that slide. You just need to know like a couple different things. That's it. So for example, in histidine, so first of all, there's histidine reaction. Basically, they're saying that histidine gives you glutamate. Okay, that is the reaction. What's important to know is that a substrate in this pathway is one of the substrates in this pathway, excuse me, is formamino glutamate, FLGLU. Okay, and that is significant because that substrate, in order for it to give you glutamate in the end, it needs tetrahydrofolate. Okay, so if someone has deficiency in tetrahydrofolate, that would increase the FLGLU. If we don't have tetrahydrofolate, that means the reaction from FLGLU to glutamate can't happen, and that would give you an accumulation of FLGLU. And what's good about this amino acid, formamino glutamate, is that it's excreted in the urine. So an excretion test to diagnose someone with folate deficiency is to see if there's FLGLU in the urine. So if there's someone that has high FLGLU in the urine, that means they have folate deficiency because they can't produce glutamate in the end, and they don't have folate, THF or tetrahydrofolate. Okay, and that's all you need to know. You guys don't need to worry about the diagram. You guys don't need to worry about those little enzymes. Those are not important. This is exactly what you guys need to know. Okay. Now folate. We talked a lot about folate and what it does. So what's good about this slide is right now there might be some stuff that you guys don't know, but again, over the next 4 or 5 lectures for MOL, you would get those concepts because they will be repeated a lot. Okay. So first of all, tetrahydrofolate is made. So let's start from here. So tetrahydrofolate is made from folate in a 2-step NADPH requiring reaction. Okay, so we get THF by two steps that have NADPH, and that is catalyzed by an enzyme called dihydrofolate reductase. Okay, so I'm gonna write it here because I think it's kind of significant. Dihydrofolate reductase. Okay, so that's how we get to THF. Now this THF is a carrier of a carbon. Okay, and that carbon group can be attached to N5, N10, or N5, N10. Okay, now based on which group it attaches to, that group will be responsible for a different reaction. Okay, for example, so an N5 methyl THF is used for a reaction from homocysteine to methionine. Okay, and we talked about it in the pathway above, where there's homocysteine goes to methionine by the help of B12, that's one cofactor, and by the help of tetrahydrofolate. Which one? The N5 tetrahydrofolate. Okay, now for an N10 formal THF, that one is used for synthesis of purine rings. Okay, you guys will talk about purine and pyrimidines in later lecture, but all you have to know is N10 is for purine ring, and N5, N10 is for the synthesis of DTMP from DUMP. Okay, that's all you need to know. Just the N5 is for pathway, the N10 for this pathway, and N5, N10 for this pathway. Okay, now when we get folate and vitamin B12 deficiency, both of these, we get something called megaloblastic anemia, which is part of macrocytic anemia. You guys will talk about it in HLS next 2 weeks, inshallah. Okay, so when there's deficiency in those 2 enzymes, folate and vitamin B12, we get megaloblastic anemia. Okay, because and why does that happen? Because there's decreased availability of purines and DTMP for DNA synthesis. Because we don't have folate here, we can't go through with this reaction, or this reaction, or this reaction, which means we won't be able to produce purines, and we won't be able to produce what is needed for DNA synthesis. Okay, so all you have to know is folate and vitamin B12 deficiency gives us a megaloblastic anemia. Okay, now if you talk about V12, vitamin B12 deficiency only, we'll talk about we'll have different symptoms and diagnostic features. So for vitamin B12, it's needed for two different reactions. Okay, it's needed for the reaction again that we talked about from homocysteine to methionine. Here it needs again tetrahydrofolate and the B12, and it's needed it's needed to break down odd number fatty acids. If you guys remember, that was in fatty acid synthesis or degradation. You guys talked about it in the beginning of the semester. So we would need vitamin B12 to break down fatty acids, odd number fatty acids, and to break down some amino acids, which are those four. And I have a different mnemonic for it. We'll talk about it in the last slide. Okay. So you guys need B12 for those different enzymes. Okay. For methionine synthase, which gives you methionine from homocysteine, and for methylmalonyl-CoA mutase, which breaks down odd number fatty acids and the four different amino acids that we talk about later into succinyl-CoA. Okay. Now I think it's kind of important to know that vitamin B12 is also called cobalamin. Okay. So every enzyme we talked about, every cofactor we talked about, there's around 2 names for it, and you guys have to like get yourself used to knowing what those names are. Okay. So when there's vitamin B12 deficiency only, not folate, we get neurological manifestations. So we get accumulation in fatty acids, which makes sense because here we can't break them down. And that would give us neurological manifestation. Neurological manifestation could be confusion, it could be peripheral neuropathy. They can't feel the ends of their limbs, their hands, and their toes. Okay. Here again, so B12 deficiency results in those two. And so tissues that would need N5, N10, and N5, N10, and N5, N10 THF are the ones required for DNA replication. So when they don't have vitamin B12, when they don't have folate, when we don't have folate, it would give us anemia. Okay, and we talked about it in the slide above. When there's only a B12 deficiency, that means we can't break down those fatty acids, or we can't make methionine from homocysteine. We get tingling in the hands and feet and neurological defects. Okay. So again, here because there's low vitamin B12, there's low tetrahydrofolate, and here there's low B12. That means there's high methylmalonyl-CoA and high odd number fatty acids. Okay. Now, for the last part of the lecture, there's cofactors that you guys talked about in MOL 1, which are TLC for Nancy. TLC for Nancy. And those enzymes are needed for pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, which you guys talked about in MOL 1, and it's needed in branch chain keto acid dehydrogenase. Okay, which we talked about up there as in I love valine one, the BCAAs. So it's needed for the BCAAs reaction, and you guys will talk about it later. But again, I love valine, those three enzymes need those cofactors, TLC for Nancy, which are thiamine pyrophosphate or thiamine B1, lipoic acid, CoA or B5, FADH2 or B2, and NADH or B3. Okay. So again, it's just branch chain amino acids. Those three amino acids require those 5 different cofactors. Okay. And for the last part of the lecture, again, we talked about how there's the odd numbered fatty acids get broken down by B12, vitamin B12, and there's 4 different amino acids that get broken down as well, and those are methionine, isoleucine, threonine, and valine. And I used to remember them by MITV. So there was a channel that I used to watch when I was a kid. It's called MTV. I just added the MI, so I made it MITV. And those are the 4 amino acids that are part of the propyl-CoA pathway that can convert to succinyl-CoA. Okay. And you guys will learn about it again later, but for now, this is the basics of what you guys need to know. Okay. Do you guys have any questions? I only have 3 questions left. So we'll just go over them quick, and then we'll give the mic to Amr to finish the lecture. I'll just go over it. A 10-year-old girl is brought to a pediatrician for a routine examination. The child is thin and has long fingers and toes, dislocated lenses, and mental retardation. Enzyme studies show an inability to convert cysteine to cysteine by cysteine B synthase. Newborn screening would have been able to identify the metabolic disorder present in this girl through the measurements of an increased serum concentration of which of the following amino acids. So all you guys need to know from this whole slide is this. Okay, so we cannot convert homocysteine to cysteine. Okay, and what is the reason for that? Like, there's an increased concentration of something that gives us.
Someone said B. They're saying B.
B? Yeah. So B is the correct answer. So it's pretty straightforward because we said methionine gives you homocysteine. So when we cannot go to cysteine, that means we have a lot of methionine. Okay, that is exactly why. So it's in the pathway up there. Okay. So now a 5-month-old infant who has been experiencing lethargy, recurrent vomiting, respiratory distress, and muscular hypotonia is brought to the emergency room near comatose. Lab results indicate severe metabolic ketoacidosis associated with an extreme accumulation of methylmalonic acid in the blood. Negative findings for pernicious anemia or other hematological or neurologic symptoms of cobalamin deficiency indicate the infant is suffering a defect in which of the following enzymes? So which of the following enzymes need cobalamin as a cofactor? That's basically the whole question. What do you guys think?
Is it C?
Yeah, it is C. So again, so C, this is the enzyme we talked about here, methylmalonyl-CoA, and that needs a cobalamin. So when there's cobalamin deficiency, that means there's something wrong with that pathway. So that enzyme is not working. Okay, so a 13-year-old boy is admitted to the hospital due to flank and urinary pain. Analysis demonstrate presence of kidney stones. Those stones were composed of calcium oxalate. Family history revealed that the boy's father and mother had similar problems. Degradation of which of the following could be the reason behind this incident? So which of the following amino acids basically can be broken down into oxalate, which give us kidney stones? That is the question.
It's E, right?
Sorry?
It's E?
Yes, it is E. Okay, well, that will be all for me today. If you guys have any questions, I'm here. I left my my contact info at the top here. And good luck. Thank you.
Thank you so much. You saved us, man.
Thank you.